So hello, I'm Bill Eldridge. I am going to be presenting VHH as building blocks for immune and tumor cell targeting. So what I'm going to do today is to introduce Isageneca to you. Also, I have a case study to present to you on our biospecifics platform and our sort of plug and play take on biospecifics. And then, having shown you a little bit of preliminary data on this, I'm then going to give you some summary and conclusions. And hopefully what you'll take away from this talk, apart from the case study itself, is what I've put up here at the top of the slide, which is that the VHH are the most clinically validated and accepted next generation antibody format at the moment. That Isageneca's Lambda library, which is a VHH synthetic library, can be rapidly mined and formatted to create these bi- and trispecific antibodies. And of course, because of their modular nature, VHH can be applied to numerous therapeutic formats. I'm just going to talk about bispecifics and trispecifics today, but there are a range of other applications, and I'll briefly cover those in the following slides. So just to introduce Isageneca to you then, we are based in Cambridge at the Chesterford Research Park, and believe it or not, Isageneca genuinely is based in this mansion at Chesterford Research Park. So we find ourselves in these very opulent surroundings, and very nice they are too. And this on the background, this is one of our labs. We have a number of labs at the Research Park. So as I mentioned already, Isageneca develops the Lambda VHH platform. This is a highly versatile small format antibody platform, and we are using these antibodies to generate next generation biotherapeutics. We have a really highly experienced team, and this team is helping partners to take their therapeutic candidates right the way from discovery through to IND. We have a track record of delivering assets through collaborative partnerships. In fact, we have a couple of Isageneca-derived and discovered candidates going through clinical trials right now. So there's one in phase 1 and one in phase 2 at the moment. So a little bit about me. I'm Bill Eldridge. I'm the current CSO of Isageneca. I've got over 20 years of experience in peptide and antibody discovery and development. And in fact, I'm a founder member of Isageneca. I am one of the inventors of Isageneca's core display technology, core discovery technology, called CysDisplay. And this allows us to interrogate antibody libraries or peptide libraries even of 3 to 4 orders of magnitude greater than is possible with more standard or traditional display technologies such as phage display. I actually left Isageneca around 10 years ago and then became CSO of a small biotech company looking at cysteine-constrained peptides. But I've recently rejoined about a couple of years ago as CSO to drive the VHH-based discovery and development platforms that we have at the company. So for those of you that aren't aware of VHH, I'm just going to briefly describe some of their sort of key characteristics. They're very small. They are antibody fragments, so they're roughly around 15 kilodaltons in molecular weight. They are very biophysically robust. That's one of the standout features of VHH. And because they're small, one would expect them to be cleared very quickly, renally, in the body. But of course, because of their modular nature, and you can daisy chain multiple VHH together to produce multi-specific molecules, you can actually produce a tunable half-life. So Isageneca, for instance, have a panel of plasma protein binding VHH, which do just that. They bind to albumin, a specific part of albumin to ensure that they don't compete with FcRn. And therefore, we can tune the half-lives of these molecules. There are a number of other ways of doing this as well. One can add fatty acid, for instance. And all of this is possible through relatively simple protein engineering techniques. And because of the modular nature of these antibody libraries, they have standardized N and C terminal ends. It allows us to be very, very flexible and versatile with the VHH products. Classically, VHH are characterized by being able to target so-called cryptic epitopes. They have extended CDRs, which allow them to penetrate into small pockets, which isn't naturally possible with normal traditional antibodies. Their small size facilitates tissue penetration. And as I've mentioned already, you can generate multivalent versions of these antibodies to produce either avid binding moieties or bispecific or trispecific moieties. And that then also allows for formatting for payload delivery. Now, of course, all of these characteristics are true for all VHH, and those are those derived from the camelid family. But Isogenica's lambda format allows us to produce antibodies that have reduced CMC liability motifs. The whole concept of our synthetic library is to have reduced liability further down the line, so that once you've discovered your antibody, you then don't have to go back to the drawing board and start removing potential sites which will cause you problems further downstream, either with regard to CMC, but also with freedom to operate as well. So for those of you that aren't familiar with the VHH format, VHH are derived from members of the camelid family. And camelids, yes, do include camels, but they also include animals such as llamas and alpacas. And all of these animals have a subset, a significant subset of antibodies which do not contain a light chain. So a light chain which is common in other mammals such as humans and mice, are not present in this subset of antibodies in the camelid family. And that means that the variable domains, the N-terminus of the antibody, do not require a light chain to pack against. And that allows them to be used as single modules. So if they don't require a light chain to pack against, you then have an individual molecule that can be modularized to produce bind tri-specific molecules, amongst other things. And that gives us the flexibility, as shown in this paper by Yang et al., that we can generate bivalent molecules or bispecifics, and we can even fuse these VHH to other therapeutic molecules. For instance, here we've got a single chain, but that could be a whole antibody. Another example is fusing them to cytokines. So a classic example of that is fusing a cancer cell targeting moiety to IL-2 to enhance cell killing via the immune system. You can also, through protein engineering and protein chemistry, fuse the VHH to non-biologic drugs as well. And so perhaps that would be a toxic payload. Isogenica ourselves, we have a very interesting collaboration ongoing with the University of Hull, Professor Steve Archibald at the University of Hull, where we are looking at using these molecules for radioimaging for disease. Again, their small size, in this case, facilitates rapid clearance, and their high affinity and specificity for their target allows us to image, in this case, cancer cells really, really clearly. It's very nice. But of course, you can also, because of their potential limitless flexibility, you can also modularize virus particles and make virus particles target particular cell types. You can use them for payload delivery, targeting and payload delivery. So we've got a couple of examples here where you're delivering a potential drug molecule or a nucleic acid. and so on and so on. The more you can think of, the more you can do with these potential biotherapeutics. As I mentioned at the beginning of this talk, VHH single domains are the most accepted next generation antibody formats. And Kablivi or caplacizumab is the first FDA-approved single-domain antibody therapeutic in this format. But there are a range, and I've just sort of highlighted a few examples of VHH-based therapeutics that are in the clinic right now. And in fact, there's the first CAR-T, VHH-based CAR-T system is this one from Legend and Janssen, and this has gone to BLA quite recently. So there are a range of potential applications. You can see they're not just used for inflammation, but in fact in the treatment of cancer, both hematologic, but also solid cancers as well, and a range of other really interesting applications. And there are a range of formats as well. So you have bispecifics, you have CAR-T applications, you have trispecifics here from Harpoon. This is a VHH, a single chain fusion. And this is a really interesting one from Lava Therapeutics, who are developing VHH for gamma delta T cell applications as well, which is a really, really exciting area. And there are numerous formats of these. There can either be monomers or bispecifics, half-life extended versions as well, and of course, CAR-T. And this, as I mentioned, this isn't an extensive table here. It just sort of gives you a snapshot of the potential applications of VHH as therapeutics. So I'm now going to move on to talking about some of the work that we've been doing at Isageneca using VHH. I've already mentioned that you can format them together as multi-specifics, and they do this far more readily than is possible with, for instance, a single chain format, which requires 2 IG domains at a time, and are associated with problems with developability in that format. With a VHH, we could produce bivalent molecules very rapidly using our cassette-based system. And in fact, I'm going to show you some examples of trivalent molecules that we've generated in the same manner. And with that, of course, you can gain improvements in affinity through avid binding. But if you produce a bispecific or trispecific molecule, you can also gain selectivity and possibly prevention of escape. So a classic example of this is HER2-positive breast cancer, which can escape Herceptin treatment via signaling through HER3. Now, if you were to produce a bispecific or trispecific molecule that targeted both HER2 and HER3, you could overcome that HER3 escape. We have a conserved scaffold, which allows the standardized manipulation of these molecules into expression cassettes. And I'll show you a little bit more about that in a second. But of course, it also opens the possibility for other potential protein chemistry approaches where we can actually fuse non-biologics onto the end of a VHH, as well as perhaps repurposing an existing antibody, for instance, introducing a second specificity onto an antibody. And there are a range of other applications as well. So at Isageneca, we have very large VHH libraries. We use both cis-display and phage display to discover our tumor-associated antigen targeting moieties. We also have a panel of albumin binding VHH as well. These all target the domain 2 on albumin to ensure that they don't compete with FcRn. And we also have a range of T-cell targeting moieties as well. And all of these exist in a single cassette-based system that allows us to rapidly clone out tumor targeting VHH into either a bispecific or trispecific molecule format. We then have the ability to express these molecules in-house, and plus perform a range of developability assessments within house, including SEC and TM, TAG, and a number of other assessments associated with stability, etc. On top of that, we can also assess these molecules for function or potency. And I'm going to show you some examples of an early panel of PD-L1 binding VHH in this format that do just that. I should mention also that our albumin binding moieties all bind protein A, which again greatly facilitates downstream processing in CMC. So let's start with our PD-L1 targeting moieties. We have a range of these. I've just shown you a snapshot here, but the monomers themselves all bind to PD-L1. And what I'm showing you here in this top left-hand graph is an antagonism assay, where we're looking at competition between PD-L1 and our VHH binders, and their inhibition of the interaction of PD-L1 and PD-1. And we've used a clinically approved antibody, made a fab of that antibody to demonstrate this as well. So our black line here is the PD-L1 antibody, and our PD-L1 VHH as monomers perform very well in inhibiting the interaction of PD-1 and PD-L1. We have a range of epitopes, as you can see. So one of our candidates is actually a partial inhibitor of PD-1 and PD-L1, whereas the other three here completely inhibit and appear to do so at a greater potency than is evidenced by the commercially available PD-L1 fab. The monomers all bind to PD-L1 expressing cells. We're using the MDA MB231 breast cancer cell line here, which is positive for PD-L1. You can see nice titration curves for each of these clones. And then as bispecifics, as we format them into PD-L1 targeting with a CD3 targeting moiety, they still retain their function as cell binders. So the commercial antibody fab is still binding absolutely fine. We have a negative control lysozyme version of this. So they have lysozyme targeting VHH coupled to our CD3 targeting moiety, which of course does not bind to the cells. And then our panel of PD-L1 targeting moieties, which are retaining their function, possibly with the exception of this one, PD-L110, which shows a little bit of a loss, but other panel members are absolutely unaffected by this, as you can see. So just to introduce our albumin binding panel very briefly, all of these albumin binders are cross-species specific. They bind to human, cynomolgus, and mouse albumin. They are domain 2 specific. Again, that's very important to ensure the half-life of your half-life extended moiety, to ensure that it doesn't compete with FcRn. And as I mentioned earlier, all of these VHH bind to protein A, and that greatly facilitates purification. And you can see that these molecules can either be fused to the N-terminus of another biotherapeutic and retain function, or they can be fused between two different moieties and still retain their function. So, you know, the affinity for albumin is unaffected by the position. That's really important when you're fusing these, trying to use these in a sort of flexible way. And our albumin clones all perform in the same way and are unaffected by position, which is very useful. So I'm now going to show you some preliminary data from the production of both bispecific and trispecific cell targeting moieties. So the blue line here is our lysozyme binding negative control. This is a lysozyme specific VHH, and that is fused to our CD3 targeting moiety at its C-terminus. As you would expect, you would not expect to see cell killing as you increase the concentration of bispecific here. With a VHH that is not targeting specifically, you would not expect to see cell killing, and it's a great relief to see that that is the case here. We have our commercially available, we have a single chain version of the commercially available anti-PD-L1 antibody here in the black line. And again, this is fused to our CD3 targeting moiety here. And you can see really quite spectacular efficacy, as one often does from this sort of bite-like format. In or around the single picomolar range. And then we have our PD-L1 bispecific, again in the same format as the single chain version, except in this case the PD-L1 targeting moiety is a VHH. And then finally we have the trispecific version of that, which is our PD-L1 VHH fused to our albumin VHH, and again fused to the CD3 targeting moiety as well. And you can see, as we look through the potencies of these individual molecules, they're all very, very close to the original BiTE, and that the addition of an albumin targeting moiety does have a mild impact on the potency of the resulting molecule, but you're still in the single-digit picomolar range. So it's really quite remarkable. These VHH were recovered directly from our libraries. We didn't perform any affinity maturation on them. We merely performed these selections and then introduced them into our bi- and trispecific format and then tested them for cell killing. I've mentioned the other panel members here as well. They're called 1, 4, and 36. And again, in each case, we see really remarkable potency in each case. We do see a slight effect on the addition of the albumin binding moiety, but again, we're still in that lows in the other two cases, just into the double-digit picomolar efficacy range, but really quite spectacular efficacy as well. And this is just to give you an idea of the flexibility and versatility of this system. So we have a range of PD-L1 binding VHH. We can drop them into a cassette-based system, express them, analyze them, and test them for function very rapidly. To ensure that the cell killing that we were observing was due to activation of T cells, we were also able to measure the upregulation of T cell activation markers. So CD25 and CD69 are classic T cell activation markers. And so from the same assay, we were able to observe upregulation of these markers using each of our candidate molecules here, as well as the anti-PD-L1 antibody-based format. And then, of course, we've got our negative control lysozyme binder going along the bottom there. Again, obviously not activating T cells and not resulting in cell killing. So hopefully there I've shown you that we have a very flexible cassette-based system that allows us to analyze tumor-associated antigen targeting moieties very rapidly in our expression cassette system. That the Lambda antibody library facilitates this by having standardized termini at the N and C termini of the VHH, which allows us to sort of literally plug and play individual molecules into the expression cassette system. We have automated high throughput systems for expression and purification and so on for these molecules, which speeds up the whole process. And on top of that, we have the capability to demonstrate both efficacy and developability. And our libraries are producing really diverse VHH antibodies with a range of antagonism profiles implying different epitopes. Now, of course, today I've just concentrated on the bispecifics and multispecifics. It plays very well into our capabilities. But there are a range of other applications for VHH as biotherapeutics. I mentioned at the top of the slide deck that the first VHH-based CAR T has gone for BLA. Their small size allows for shorter genes and therefore ease of manufacturing and no chain pairing, which improves the stability of the molecule. And of course, it allows one not simply to be tied to a single tumor-associated antigen. You could potentially have 2 or potentially even 3 tumor-associated antigen targeting moieties on your CAR T. They can be used as direct therapeutics, and Cablivi is an example of that, and there are many others following through. And just as interestingly, they can be applied to antibody drug or radionuclide conjugates, and we ourselves have a collaboration directed at exactly that kind of approach. So their modularity and flexibility facilitates all of these applications, and that's why VHH have been developed and are being developed as therapeutics and as next generation therapeutics as well. So Isageneca have a high throughput screening system that allows us to interrogate our very large and CMC liability reduced libraries. We have a range of capabilities, not only in antibody discovery, but also in functional assessment as well. And we have a range of skills ourselves that allows us to take antibodies from discovery right the way through to IND. And we're leveraging this platform to build this internal discovery pipeline of our own, as well as working with partners to develop their own therapeutics. And with that, I'd like to close and await any potential questions. Thank you.